Hello everyone, my name is Philip Vines and I am an assistant professor of turfgrass breeding at Rutgers University. In this presentation, I will be discussing a recent study we conducted in comparing the relative virulence among Magnaporthiopsis species on hard fescue and Kentucky bluegrass host. Summer patch is a destructive root disease of hard fescue and Kentucky bluegrass turf. Initial disease symptoms included scattered, small, crescent-shaped to circular patches, as shown here in Figure 1. Infected plants initially turn chlorotic and then tan to straw-colored as leaves wilt, turf thins, and ultimately plants die. Patches range in size from 5 to 30 centimeters in diameter and may be recolonized by resistant grasses or weeds in the centers, which can often create a frog eye or donut-shaped appearance. The fungal pathogen Magnaporthiopsis poe has long been recognized as a causal agent for summer patch disease. However, two new fungal species, Magnaporthiopsis cynodontis and Magnaporthiopsis meyeri, have recently been identified from diseased roots of hard fescue and Kentucky bluegrass turf exhibiting typical summer patch disease symptoms. The objective of this research was to investigate the disease virulence among isolates of Magnaporthiopsis cynodontis, Magnaporthiopsis meyeri, and Magnaporthiopsis poe to better understand which of the pathogens, and more specifically, which isolates affect hard fescue and Kentucky bluegrass turf the most. With respect to the inoculations, for this study, we used 4 isolates of Magnaporthiopsis cynodontis, 11 isolates of Magnaporthiopsis meyeri, and 11 isolates of Magnaporthiopsis poe. Inoculum was prepared by growing each isolate on sterilized oat grain for 30 to 40 days at 20 degrees centigrade. Inoculations were carried out in 3.8 centimeter diameter by 20 centimeter long containers. The containers were filled to a level 3 centimeters below the top of the containers with sterilized calcined clay. Five inoculated oat grains were then added to provide an inoculum layer in the soil profile. An additional 1.5 centimeter deep layer of calcined clay was added over top the seed layer. Each container was seeded with beacon hard fescue or A1620 Kentucky bluegrass at a rate of 9.7 grams of seed per meter square. A 1 centimeter deep layer of calcined clay was added over top the seed layer. Control units were constructed in the same manner, however the oat grain in those was not inoculated with the fungus. Plants were established in a mist chamber for 30 days before transferring to light and temperature controlled growth chambers. There were 2 environmental conditions in this study. Firstly, we had a warm environment that had 12-hour photo periods with day temperatures of 29 centigrade and night temperatures of 25 centigrade. We also had a cool environment that had a 12-hour photo period with day temperatures at 24 degrees centigrade and night temperatures at 20 degrees centigrade. Both environments were maintained at 70% relative humidity. Plants were watered 2 times daily, misted 4 times daily, trimmed weekly at a height of 5 centimeters, and re-randomized and fertilized bi-weekly. Plants were maintained for 13 weeks and 15 weeks for experiments 1 and 2 respectively. In consideration of the experimental design, disease evaluation parameters, and data analysis of this experiment, the treatment factors in the study included the fungal isolate, the host plant species, and the environmental condition. There were 27 levels of fungal isolate, 2 levels of host plant species, and 2 levels of environmental condition. Treatments were arranged as a 27 by 2 by 2 factorial in a randomized complete block design with 3 replications for each treatment combination. Plants were evaluated weekly for percent disease and disease intensity in both experiments. Percent disease was visually estimated for all plants in a given container, and disease intensity was assessed using a 1 to 5 visual rating scale, where 1 represented healthy plants, 2 represented plants that were slightly chlorotic or necrotic, 3 represented plants that were moderately chlorotic or necrotic, 4 represented plants that were severely chlorotic or necrotic, and 5 represented plants that were dead. Disease severity index was calculated for each container as percent disease times the disease intensity. The area under the disease progress curve was calculated based on disease severity index. The area under the disease progress curve data were subjected to an analysis of variance using the Glemmick's procedure of SAS. Now for the results and discussion. The area under the disease progress curve was significantly affected by the four-way interaction of experiment, by environment, by host, by isolate. The mean area under the disease progress curve was smaller in experiment 1 than experiment 2. The difference in this mean area under the disease progress curve difference between the two experiments could be attributed to a difference in length of study and evaluators. Experiment 1 was concluded 13 weeks after establishment, whereas experiment 2 was concluded 15 weeks after establishment. Experiment 2 was continued for an additional 2 weeks to increase disease symptom expression. In addition, the plants of the two experiments were evaluated by 2 different evaluators, and it is possible that this also contributed to the observed differences in the area under the disease progress curve between the two experiments. Now, when we look at the experiments independently, first we'll look at experiment 1. The Kentucky bluegrass had a greater area under the disease progress curve than hard fescue in experiment 1, and a greater AUD-PC was observed in the warm environment compared to the cool environment. Area under the disease progress curve was greater on the hard fescue in the warm environment as compared to the cool environment. Likewise, for Kentucky bluegrass, area under the disease progress curve was greater in the warm environment than the cool environment. On hard fescue in the cool environment, Magnaporthiopsis poe isolates incited greater area under the disease progress curve than Magnaporthiopsis cyanodontis and Magnaporthiopsis Meyeri festucii isolates. The Magnaporthiopsis poe isolates C3, P5, and Will A3 incited significantly greater disease symptoms than the untreated control, and that is shown in figure 2A. Magnaporthiopsis poe isolates also incited greater area under the disease progress curve on hard fescue in the warm environment compared to M. cyanodontis and M. Meyeri festucii isolates. M. poe isolates 37S, C3, FF SP12, Oak A5, and P5 incited significantly greater disease symptoms than the untreated control, and that is shown in figure 2B. On Kentucky bluegrass in the cool environment, M. poe isolates incited greater area under the disease progress curve than M. cyanodontis and M. Meyeri festucii isolates. The M. poe isolates 37S, Bow B24, HF22, Lisa 9, Oak A5, and Will A3 incited significantly greater disease symptoms than the untreated control, and that is shown in figure 2C. On Kentucky bluegrass in the warm environment, M. poe isolates incited greater area under the disease progress curve than M. cyanodontis and M. Meyeri festucii isolates. The M. poe isolates 37S, Bow B24, Lisa 9, Oak A5, STD A7, and Will A3 caused significantly more disease symptoms than the untreated control, and that is shown in figure 2D. Other isolates incited disease symptom development. However, they were not statistically different from the untreated control, which had no disease symptoms. Nonetheless, M. cynodontis and M. Meyer festuci isolates could play a role in summer patch disease development in hard fescue and Kentucky bluegrass turf, and should be considered in future studies for potential synergistic interactions with M. poe. Now, looking ahead to Experiment 2, like Experiment 1, a greater area under the disease progress curve was observed on Kentucky bluegrass compared to hard fescue in Experiment 2. A slightly greater AUDPC was observed in the cool environment compared to the warm environment. Area under the disease progress curve was greater on hard fescue in the warm environment than the cool environment. For Kentucky bluegrass, area under the disease progress curve was greater in the cool environment than the warm environment. On hard fescue in the cool environment, M. poe isolates incited greater AUDPC than M. cynodontis and M. Meyer festuci isolates. M. poe isolates 37S, C3, FFSP1-2, Lisa 9, and P5 cause significantly more disease than the untreated control, and that is shown in Figure 3A. On the hard fescue in the warm environment, M. poe isolates incited greater area under the disease progress curve than M. cynodontis and M. Meyer festuci isolates. M. poe isolates 37S, C3, FFSP1-2, Lisa 9, P5, and STD-A7 incited greater disease symptom development than the untreated control, as shown in Figure 3B. On Kentucky bluegrass in the cool environment, M. poe isolates incited greater area under the disease progress curve than M. cynodontis and M. Meyer festuci. M. poe isolates 37S, C3, FFSP1-2, Lisa 9, Oak A5, P5, and STD-A7 had more disease symptom development than the untreated control, as shown in Figure 3C. On Kentucky bluegrass in the warm environment, M. poe isolates incited greater area under the disease progress curve than M. cynodontis and M. Meyer festuci isolates. The M. poe isolates 37S, C3, FFSP1-2, Lisa 9, Oak A5, P5, STD-A7, and M. cynodontis isolate D29740-4 incited significantly more disease than the untreated control, as shown in Figure 3D. As was mentioned based on Experiment 1 results, M. poe isolates incited the most disease in Experiment 2. However, isolates of M. cynodontis and M. Meyer festuci also caused disease symptoms on hard fescue and Kentucky bluegrass, and these isolates should be considered in future studies to determine the specific roles they play in summer patch disease development. So with that, a look at conclusions and future work. In this study, we investigated the virulence of 3 Magnaporthiopsis species that have been associated with summer patch disease symptoms in both hard fescue and Kentucky bluegrass turf. Our results indicate that M. poe is more virulent to hard fescue and Kentucky bluegrass than either M. cynodontis or M. Meyer festuci. However, M. cynodontis and M. Meyer festuci did incite disease symptoms on both turf species, and these findings suggest that M. poe contributes mostly to summer patch disease in hard fescue and Kentucky bluegrass. But these findings also underscore the activity of M. cynodontis and M. Meyer festuci on these plants as well. Future studies should consider the potential synergistic relationship among these fungi and investigate the impact that multiple species would have together on these host plant species. To complete this study, other important data parameters such as root and shoot weights will need to be considered. Those data have been collected for both experiments in this study, and appropriate analyses are being done to make final conclusions regarding the virulence of M. cynodontis, M. Meyer festuci, and M. poe on hard fescue and Kentucky bluegrass turf. With that, we'd like to acknowledge our greenhouse support, Mr. Joe Florentine and Jeff Akers, as well as our lab support, Dr. Ning Zhang, Dr. Jing Lu, and Patrick Perdon. We'd also like to thank our funding from the New Jersey Turfgrass Association and the Rutgers Center for Turfgrass Science. My contact information is listed here if anyone needs to reach out to me for questions. Thank you.